talking about Chapter 12, the central nervous system. So, the central nervous system consists of the brain and spinal cord, and we approach it from a perspective of evolution. So, the process of forming this brain and spinal cord, especially the brain and the head, is known as cephalization. That's the evolutionary development of the anterior portions of the central nervous system. This has resulted in an increased number of neurons and the highest number of neurons, organization of neurons, has been reached in the human brain. I would caution you to remember that this was written by humans, so animals with bigger brains, although they're not as convoluted and not as folded as ours, might argue. Anyway, another joke. So embryologically, the brain and spinal cord start out as a neural tube. The neural tube's anterior end expands and then constricts and forms three vesicles. Those vesicles are known as the proencephalon, the mesencephalon, and the rhombencephalon, also called forebrain, midbrain, and hindbrain. And the posterior end is going to become the spinal cord. Those primary vesicles, three primary vesicles, become five secondary vesicles. So the forebrain is going to become the telencephalon and the diencephalon. The midbrain is not going to divide, but the hindbrain is going to divide into the metencephalon and the myelencephalon. Telencephalon is going to give us our two cerebral hemispheres, which together will make up the cerebrum. The diencephalon is going to become the epithalamus, the thalamus, the hypothalamus, and the retina. The mesencephalon is the midbrain. The metencephalon becomes the pons and the cerebellum. The myelencephalon becomes the medulla oblongata. And the central cavity of that neural tube will become the ventricles within the brain. So we can follow this from the neural tube to the adult neural regions. Brain grows faster than the surrounding membranous skull, so it has to fold to occupy the available space. The forebrain moves toward the brain stem, the midbrain pons, medulla oblongata. Central hemispheres double back and envelope the diencephalon, and the midbrain increases and folds to increase surface area. So that's kind of a choreographed type of movement and migration that allows this, what would otherwise be a very long structure, to fold back on itself, develop folds on the surface, and become the brain that we are used to. And that adult brain has four main regions: the cerebral hemisphere, the diencephalon, the brain stem, which is will divide into the midbrain, pons, and medulla, and the cerebellum, which kind of hangs out in the back. So here's the brain you're probably used to looking at. It is a highly folded structure to get all of those neurons in there, and it's that increased surface area, this whole thing being covered with cell bodies for the most part, communicating with one another, integrating information, as we discussed a little bit last week. So gray matter are the short, non-myelinated neurons and cell bodies, whereas white matter is myelinated and non-myelinated axons. The basic patterns we find for this white and gray matter is in the brain and spinal cord. Gray matter is on the external part, white matter is internal. That will switch when we get to the spinal cord itself. There, the gray matter will be internal and the white matter will be external, because most of our thought and communication, our integration, interpretation, goes on the surface of our brain, the cerebral hemispheres, and that's communicated through the white matter tracks to the rest of the body, whereas most of the integration in the spinal cord is going to be close to the central nervous system, around the neural tube, and then it's going to communicate outward to the body. The brainstem has additional gray matter structures within it, cell bodies within it, we call nuclei. They're groups of cell bodies within the white matter. Some of those are named, some of them we'll be talking about later on when we talk about functional regions of the brain. The cerebral hemisphere and the cerebellum contain an outer layer of gray matter called the cortex. So we've got the cerebral cortex and the cerebellar cortex. They also have nuclei within them as well. The ventricles are fluid-filled chambers that are continuous to one another in the central cord of the spinal cord. Those chambers are filled with cerebral spinal fluid, and they're lined by epidermal cells. Remember those neuroglial cells from last week. They have a pair of lateral ventricles. They're large and kind of C-shaped chambers located deep beneath each hemisphere, and the two pairs are separated by a membrane called the septum pellucidum. Each lateral ventricle is connected to the third ventricle through the intraventricular foramen. That third ventricle is in the diencephalon. Third ventricle is connected to the fourth ventricle through a structure called the cerebral aqueduct. The fourth ventricle is in the hindbrain. It's continuous with the central canal of the spinal cord. There are three openings connecting the fourth ventricle to the subarachnoid space, which surrounds the brain. There's a pair of lateral apertures in the sidewall and a median aperture in the roof. So here are pictures of what I've been trying to describe to you. Hopefully a little less abstract with the pictures than in the description. Cerebral hemispheres form the superior part of the brain. About 83% of the brain mass is in the cerebral hemisphere. It is a highly folded surface, giving us more surface area, allowing us to have more of those cells, more connections. And places that stick up a little bit in those folds are called gyri. The places where those gyri, those folds go down, are called sulci, unless they go quite deep, and then they are referred to as fissures. They're longitudinal fissure separates the two hemispheres, and the transverse cerebral fissure separates the cerebrum from the cerebellum. So here are the folds we're talking about. These parts that are folded upward are called the gyri. The inner parts of those are the sulci. And then here's the longitudinal fissure. You can see it goes quite deep. And this is a side view. Again, we've got these divided into lobes, named for the bones that cover them in most cases. And you'll get to see these when we do this in the lab. Not this week, but next week. So the hemispheres, those sulci are divided into each hemisphere into 5 lobes. Those lobes are, except for one, named for the bones that cover them. We have the frontal, the parietal, the temporal, the occipital, and the insula. So 4 of them are named for the overlying skull bones. The insula you can't see from the outside really. You got to kind of probe inside a little bit, and you'll see it here in an image in a second. So there's your brain, or a brain. Hopefully it's not yours. And you can see if we, I lost my pointer, but if we pull the region between the red and the blue apart, the frontal lobe and the temporal lobe a little bit, you can see the insula region. There's a better image of it. So the sulci that divide the lobes would include the central sulcus, which separates the precentral gyrus of the frontal lobe and the postcentral gyrus of the parietal lobe, which we'll be talking about shortly. The parietal occipital sulcus is going to separate the occipital and parietal lobes. The lateral sulcus outlines the temporal lobes. Each hemisphere is going to have 3 basic regions: the cerebral cortex, which is the gray matter on the surface, white matter internally, and then included in that white matter, but very deep in it, are the groups of cell bodies known as the basal nuclei. So cerebral cortex, white matter, and basal nuclei for each hemisphere. Cerebral cortex is our executive suite of the brain. It's the site of our conscious mind, the site of awareness, sensory perception, voluntary, motor. At least it starts there: initiation, communication, memory storage, understanding things. It's very thin, between 2 and 4 millimeters. It's composed of neuron cell bodies, they're dendrites, many glial cells, blood vessels, but no axons. Those axons would be in the white matter. And about 40% of your brain mass is in your cerebral cortex, almost half. We use functional imaging like PET and MRIs to show specific motory and sensory functions located in discrete cortical areas called domains. Higher functions are going to be spread out over many areas, so don't ever fall for the line. We only use 10% of our brain, or whatever percentage they tell you. We are using all of our brain, maybe not at every single second, but to do things that we do consciously, to maintain consciousness, requires the entire structure. We know a lot about brains unfortunately, from brain injuries that have occurred over the years. Used to be that was the only way we could get information about the brain, but now we've got some new imaging and functional tests that can tell us what's going on, not only in pathology, but also in normal brain function. So if we ask someone to look at something, a particular region of the brain will light up. This is an fMRI of the cerebral cortex. Four general considerations for the cerebral cortex. It contains 3 types of functional areas: motor areas, sensory areas, and association areas. Motor areas are where we start the control of voluntary movement. We get the idea we want to do something, that's where it originates, and then it's processed a lot, especially in the cerebellum, but it starts here. Sensory areas are where we have conscious awareness of our sensations. And then association areas are going to be the places where this black box of integration occurs. Each hemisphere is concerned with the contralateral side of the body, for the most part. So your right side does control, the right side of your brain does control the left side of your body in general, and vice versa. There is some lateralization. There is some specialization of cortical functions. So some cortical functions happen only in one hemisphere. But again, conscious behavior is going to involve the entire cortex, both sides, one way or another. So we'll start with motor areas. Most of these are located in the frontal lobe. Motor areas act to control voluntary movement. Our primary motor cortex is in the precentral gyrus. Our premotor cortex is just behind the precentral gyrus. There's a region called Broca's area that's involved in speech. It's just below the premotor area. And there is a frontal eye field within and anterior to the premotor cortex, just above Broca's area. And these are shown here in figure 12.7. Broca's area and Wernicke's area are named because of 2 anatomists who studied patients who had had strokes and had trouble with their speech. There is Bruce Willis, who is suffering from aphasia. I'm not sure what type of aphasia, but there 2 common aphasias are Broca's aphasia and Wernicke's aphasia, which interfere with how we speak. The primary motor cortex is located in the precentral gyrus of the frontal lobe. Here we find pyramidal cells, which are large neurons that allow conscious control of precise, skilled skeletal muscle movements. The pyramidal or corticospinal tracts are formed from the long axons that project down to the spinal cord. Somatotopy are all the muscles can be mapped to areas on the primary motor cortex, creating an image what we refer to as the motor homunculi. It is an upside-down character representation of the contralateral motor innervation of body regions. So I believe the next image is that. I'm sorry, my pointer's still gone, but you can see that the features are all exaggerated on the individual. And those size of those features is related to the number of skeletal muscles in those areas. So we have a lot of skeletal muscles in our face, in our hand, very little in our wrist. It does work primarily in an inverse portion. We have a similar structure on the other side we'll talk about called the sensory homunculi. But here you see the characters are drawn differently because this is where we have sensory information coming in, not motor information. The premotor cortex is going to help plan movements. It's a staging area for skilled motor activities. It's going to control learned, repetitious, and pattern motor skills. It's going to coordinate simultaneous or sequential actions. And it controls voluntary action that depend on sensory feedback. So again, Broca's area is found in one hemisphere. It is usually the left. It's a motor speech area that directs muscles of speech production. It also is active in planning speech and voluntary motor activities. Our frontal eye field is going to control voluntary eye movements. So damage to areas of the primary motor cortex is seen as a stroke, will cause paralysis on the muscle controls to that area, and the paralysis is going to occur on the opposite side of the body as the stroke did because of that contralateral control. Muscle strength, the ability to perform discrete individual movements, is not impaired. Only control over the muscle movement is lost. So, for example, damage to the premotor area controlling movement of the fingers would still allow the fingers to move, but voluntary control needed to type would not be available. Other premotor neurons can be reprogrammed to take over the skills of the damaged neuron. That requires practice, just as the initial learning process required practice. And that is the goal of physical therapy. In the cerebral cortex, we have sensory areas. They're concerned with conscious awareness of sensation. We find them in the parietal, the insular, the temporal, and the occipital lobes. There are many of them. The 8 we'll talk about are the primary somatosensory cortex, somatosensory association cortex, visual area, auditory area, vestibular cortex or olfactory cortex, gustatory cortex, and the visceral sensory area. So the primary somatosensory cortex is located on the postcentral gyri of the parietal lobe. It gets general sensory information from skin and the proprioceptors of the skeletal muscles, joints, and tendons. It's capable of spatial discrimination. It identifies the region of the body being stimulated based on where the signal comes in. So we have, again, a somatosensory homunculus, that upside down caricature, representing the contralateral sensory input areas from the body. It was over here on the screen's left. The association cortex is posterior to the primary somatosensory cortex. It's going to integrate that sensory information from the somatosensory cortex so that we can understand the thing that we are sensing. It's going to determine things like size, texture, and relationship of the objects we're feeling. So visual areas, we have a primary visual called the striate cortex located on the extreme posterior tip of the occipital lobe. It's mostly buried in the calcarine sulcus, and it receives visual information from the retina. Then we have visual association areas that surround that primary visual cortex, and they are going to use past visual experience to interpret visual stimuli, things like color, form, or movement. We know there are regions associated with recognition of faces in particular. There's a very complex processing that involves the entire posterior half of the cerebral hemispheres. Our our vision is the thing we have to teach the most, and it's the thing that requires the most of this integration. Auditory areas would include the primary auditory cortex and the auditory association area. The primary cortex is going to interpret information such as pitch, loudness, and location, while the association area is going to use that to store memories and interpret the sound that we are hearing. The vestibular cortex is in the posterior part of the insula and adjacent parietal cortex. It's aware of our balance, our position, our head in space. The olfactory is our sense of smell. Cortex is the medial aspect of the temporal lobes. It's part of a very primitive part of the brain called the rhinencephalon, which means that a lot of species have that. It's an early evolutionary development. And the rest of that is going to become the limbic system, which is an emotional system. This is where we get, this olfactory region is where we get conscious awareness of odors, and that's part of the reason that odors are such a strong trigger of emotions. Gustatory cortex is in the insula, deep to the temporal lobe. That's involved in our perception of taste, if something is good or bad. The visceral sensory area is posterior to the gustatory cortex, and it's where we get perception of visceral sensations. Upset stomach, full bladder, those kinds of things. Okay, so damage to the primary visual cortex can result in functional blindness. So if your eyes work fine, but you cannot interpret the information coming in, by contrast, individual with damage to the visual association area can still see, but they can't comprehend what they are looking at. And then we have multimodal association areas that receive inputs from multiple sensory areas and send outputs to multiple sensory areas. This allows us to give meaning to information received, stored in memory, tie it to previous experience, and decide on our actions. Sensations, thoughts, emotions become conscious. They make us who we are. It's going to be broadly divided into three parts: the anterior association area, the posterior association area, and the limbic association area. The anterior association area is also called the prefrontal cortex. It's the most complicated of the cortical regions. It's involved with our intellect, with cognition, recall, and our personality. It contains our working memory that's needed for abstract ideas, for judgment, reasoning, persistence, and planning. Its development depends on feedback from our social environment. This is one of the regions that is fully developed, the latest in life. When you hear teenagers' brains are not fully formed, it is this prefrontal cortex that is still in the process of making its connections. Posterior association area is a large region of the temporal, parietal, and occipital lobes. It's involved in recognizing patterns, faces, and localizing us in space. It's also involved in understanding written and spoken language. That would be what we call Wernicke's area. And the limbic association is part of the limbic system. It involves the cingulate gyrus, the parahippocampal gyrus, and the hippocampus. This is where we get emotional impact and makes a sense of important things to us, a very important part of establishing memories. So tumors or other lesions of the anterior association area can cause mental and personality disorders, including loss of judgment, attentiveness, and inhibitions. Individuals affected with this can be oblivious to social restraints, perhaps becoming careless about personal appearance or taking risks. Different problems arise for individuals with lesions in part of the posterior association area that provide awareness of self in space. Those individuals may refuse to wash or dress one side of the body because that doesn't belong to them in their mind. The cerebral cortex, there is lateralization of some cortical functions. That means that it's only on one hemisphere, not the other. Cerebral dominance refers to the hemisphere that is dominant for language. About 90% of humans have left-sided dominance. That usually results in right-handedness. And the other 10%, the roles of the hemisphere are reversed. The left hemisphere controls language, math, and logic. The right hemisphere is more important for visual, spatial skills, intuition, emotion, and artistic and musical abilities. However, there is no such thing as a left brain person or a right brain person. Those hemispheres are communicating almost instantaneously through fibrous tracts and functional integration. We are all using both sides of our brain. Okay, I'll stop there. And